Thanks everyone for turning up. It's a great rollout for tonight, so hopefully I can keep you engaged for the next 20 minutes or so. Talking about celiac disease. My name's James Davis and I'm a gastroenterologist. I have sort of an unusual practice. I grew up in the regions. I still travel to the regions every week where I think there's a real need for specialty care. But when I'm in Brisbane, I tend to spend more of my time engaged in research and facilitating that. What I want to speak about tonight is in three parts really. So a bit about celiac disease to start with, and the second part will be probably what most people are interested in, is where the NextVax vaccine is up to. And finally, just some things we've been trying to do here for the last 12 months at WMR. And I just want to challenge to start with probably a common misconception about celiac disease. So I'm pretty sure if I walked out to the footpath now and asked 100 people what celiac disease was, I'd probably get the response, oh, it's that thing where, you eat a bit of gluten and get a bit of diarrhea and bloating, and, if you go off the gluten, you'll feel better and there's not much else to worry about. And I think celiac disease has been caught up in the whole the gluten-free diet. And I think that's been one of the problems with the disease. But the reason why I think that's happened is when the cause of celiac disease was defined back in the 30s, it was around the time that doctors began to be able to biopsy the bowel. So the two have grown together, the cause of celiac disease and our ability to see the bowel. So this is where it all began. So this is Wilhelm Dickey. This is in the 1930s. And this is obviously a young child who doesn't look terribly well at all. So there's no fat on this child. Malabsorption was a huge problem back then, and no one knew what the cause of celiac disease was. And no one knew that gluten was a causative agent. So this is a tribute to Wilhelm Dickey, and I think it's really important to know that prior to him, the mortality rate of celiac disease was around 30%. So this is a study they refer to in this tribute, where they had 10 people. So the treatment then was rest and diet. And of those 10 people, they put 8 on a banana diet because they knew that worked. And those 8 improved, but the other 2 passed away. So celiac disease had a mortality associated with it. And I think the other thing was Dickey realised that when one of his patients came along, the young mother said, look, I think the problem with my child is when I get them to eat wheat, they become unwell. And when I take it away, they seem to improve. So what he devised was a fecal test that measured the fecal fat in the stool. And he was able to show that if you gave these children gluten, their fecal fat increased. So they began to malabsorb. And when you took the gluten away, their fecal fat levels dropped. So that was really the first test to be able to look at celiac disease. Then the next important one was when doctors came along and they were able to biopsy the bowel. Now, I wasn't around when this was used, but this is a Crosby capsule, and some people may have seen it used. But a Crosby capsule has this little part at the end, and what happens is the patient swallows that, and then by natural peristalsis, it's pulled down into the small bowel. And when on X-ray it seemed to be in the right position, they attach a little suction mechanism to the under end of that lining. And it pulls in the mucosa and cuts off the top of it, and then that's pulled back out. And then you're able to get the mucosa out of the Crosby capsule, and we're able to look at it on a microscope. So around the time that people started to realise people were malabsorbing when they were eating gluten, we also began to see the effects in the bowel of celiac disease. So the two sort of went on that journey together. But things have changed. So that was 70 years ago. And there's been a number of seminal publications since then that have changed the way we think about celiac disease. And I now don't think of celiac disease as primarily a gut disorder. I see it as an autoimmune disease, and I see it as a systemic immune disease where the gut is involved. And it's a slightly different way of thinking about it. And I think it's important to think about it that way if we're going to discuss what the therapies for celiac disease will be. So in Quesada in 1980 actually started to map the gluten protein. So before that we knew wheat was the problem, but we didn't know what in wheat was the problem. And the way to think about gluten is it's a long chain of pearls. There's hundreds of gluten proteins, and it's not the whole chain that causes the problem, it's just a small part of it. So that's a small part of the gluten protein chain. And I've drawn it diagrammatically here because the next few slides are going to be showing what the pathogenesis of celiac disease is and how it works. So if you think of this as the gluten protein, which is really quite long, and there's a small part of that gluten protein that the human body can't break down any further than that, that's the bit causing the problem. So the next thing about celiac disease that Solid and Thorsby realised was that genes are important for celiac disease. So if you have the right genetic makeup, you are predisposed to celiac disease. Whereas if you don't have that, you almost certainly will not get celiac disease. And the reason why that's important is that gene codes for this little molecule. So this is an antigen presenting cell. So antigens are things that are foreign to our body, such as viruses or bacteria. And when they get into our body, our immune system takes them up and it presents it on this little molecule. And that sends a signal to the rest of the immune system, in this case CD4 cells. And what these CD4 cells do is then they upregulate the immune system in a certain way. And one of the things we see is they upregulate and they secrete interleukin-2, which is a type of cytokine which I'll come to in a minute. So you imagine this is the gluten protein, that long chain of amino acids. It gets broken down in the gut. This small bit gets presented on this HLA molecule, and that gets presented to the rest of our immune system. So the genes are really important, and that gluten protein is also really important. So this is a pathogenesis. So the gluten protein swallowed in the gut gets broken down. Humans can only break it down to a certain extent. That toxic fragment gets taken up across the bowel. It gets presented on this molecule, on antigen presenting cells, presented to CD4 T cells, and they release cytokines. So if you think of celiac disease in this way, it is an immune-mediated disease. It is not a gut disease. So it's a lifelong immune-mediated systemic disease. The definition of celiac disease changed around 15 years ago. There is a genetic predisposition to it. You have a HLA-DQ2 or 8. We still diagnose it with small bowel biopsies and blood tests, and we know that it's gluten derived from wheat, rye and barley, and sometimes oats. So traditionally, people also think of celiac disease as being a disease of Caucasians, but that equally is not true. Wherever you find the gene, you will find the disease. So traditionally in Europe, Australia and America, but celiac disease is also found in India, it's found in Asia, and it's found in Africa. The only place I think where the prevalence is thought to be low still is Japan, because there's probably not a lot of interracial marriage. In Australia, we think the incidence is 1 in 60 for females and 1 in 80 for males. And I think the important thing here is, that's been touched on already tonight, is so about half the population is not genetically susceptible to celiac disease, while the other half is. Of those ones that are, about 1 in 20 will develop celiac disease, so 1.6% of the Australian population. But still in Australia, we're only diagnosing 1 out of every 5, so 0.3%. So 4 out of 5 people with celiac disease are still walking around and don't know they have it. And as Professor Allen indicated, the incidence of celiac disease is increasing. So as traditional diseases, so there will be physicians in this room that would have regularly treated these diseases in their younger days. But for us and younger physicians, we don't see a lot of this anymore. We have good antibiotic therapy, we have good vaccinations, measles, hepatitis A, tuberculosis, they're all on the decline. But what we are finding is autoimmune diseases are increasing in incidence. So multiple sclerosis, celiac disease, Crohn's disease, type 1 diabetes, all autoimmune diseases and all going up. The beauty about celiac disease and why it's such a lovely disease to study is we know what the trigger is. We know that gluten peptide triggers celiac disease, and it's a very well characterised disease. The others we don't really know what the trigger is. But if you can understand celiac disease and if you can develop therapies for it, it really is a large platform for moving on to treat these other autoimmune diseases. So in the United States, celiac disease seems to be doubling in incidence every 15 years. And I like this study because whenever you talk about a rising incidence of a disease, people go, well, isn't it just that we're testing more people? So what happened with this study is they got serum from 9,000 military recruits from 1950. And they pulled that out of the fridge and they thawed it, and then they tested it for celiac antibodies. And then they age and sex matched that cohort, so fairly similar people, but from 2007, so around 50 years later. And what they found was that in the 1948 cohort, when you tested that blood for celiac disease, 0.2% of them had positive blood tests for celiac disease. But when you tested the cohort from 2007, 1% had now had positive serology for celiac disease. So 5 times the incidence. And that was statistically significant. But the really interesting part of this study, I thought, and this was not statistically significant, but still impressive, that they then went back and looked and said, well, of these people, how many are still alive 40 years later? So how many have passed away? And if you looked at those that had negative serology for celiac disease in 1948, over the ensuing 40 years, 17% had died. But if you looked at those who had positive serology over the next 40 years, 50% had died from all-cause mortality. So it harks back to that original slide that when young people were passing away with celiac disease from malabsorption, that was probably the tip of it. That was the most severe form of celiac disease. But celiac disease is not present in that manner to everyone. So how do we diagnose celiac disease? Well, I think when it's obvious, it's obvious. So these days, that child wouldn't get through emergency without having celiac serology done. This is dermatitis herpetiformis, which is a skin manifestation of celiac disease, and it tests to that whole theory that this is a systemic disease, not a gut disorder. And this is enamel hyperplasia. So you can almost date the onset of the gluten-free diet in this child. So prior to going on to a gluten-free diet, you can see enamel wasn't being put down on these teeth. After a gluten-free diet was initiated, there was plenty of enamel. So when we see people in our clinic, we think now only 30 or 40% of people present with symptoms when they have celiac disease. Around 30% do not have symptoms or think they don't have symptoms at the time of diagnosis. And it may only be 6 or 12 months later when they look back and go, well, I was unwell. And it was this that was part of my celiac disease. We're finding a lot from complications, so osteoporosis or iron deficiency. And we're now actively looking for cases. We're testing first-degree relatives if someone has celiac disease, which we'll come to. These are the blood tests that we use for celiac disease, and we also now usually include their genetic test just to see if they are at risk. The blood tests are gluten dependent, so you need to be eating enough gluten for these tests to become positive. So that's why when people come and see us and they haven't had blood tests and they're on a gluten-free diet, we need to put them back on gluten. And that may be changing, but currently that's what we need to do. But if they're on a gluten-free diet, we can test their genes. And if they don't have a HLA-DQ2 or 8, there is really no need for them to have that 6-week gluten challenge to make themselves feel unwell, because the chance of them having celiac disease is remote. But the Crosby capsule has passed, and now we have more sophisticated ways and probably safer ways of diagnosing celiac disease. And we pass gastroscopes down into the second part of the bowel and take several small biopsies, which is really a very safe thing to do. These are the villi. So every 3 to 5 years, the scopes we use improve, the graphics improve. So you can actually see the villi when we go down there on high resolution. And with the really good scopes, you can actually see the blood coursing up and down the villi now. So this is someone with celiac disease. They've got no villi left. All you can see is these dilated crypts in their bowel. You can see this mucosal erythema. And this is when we biopsy it. Instead of having these nice elongated villi, it becomes very flat. And when people come and say, well, I feel well, do I really need to have a diagnosis? Can I just continue on my gluten-free diet? And I would say it's really important to do that. So if you have celiac disease and you don't know about it, or you're not treating it, you have a 3-fold risk of autoimmune disease which may not be looked for, so thyroid disease. You're at risk of osteoporosis and you're at risk of lymphoma and malignancy. We know people have a decreased quality of life, and we know, as you pointed out, and it makes sense to me, if you have a young child who doesn't know they have celiac disease and you're giving them cereal for breakfast and sending them off to school and giving them lunch, which are sandwiches, and they come home and have more gluten, they tend not to do as well. And there's a lower likelihood those kids will go on to a university degree. But by treating this and getting onto it early, you can reverse those complications. And when we were discussing before, we often see there's really 3, I think, opportunities to diagnose celiac disease in children, just as when they're 2 or 3, when they're 9 or 10, or then when they're in their late teens, because they're their 3 real growth opportunities. And if you can diagnose it then and turn it around, you often see kids almost explode in height and weight. So they may look well, but their gut is inflamed. There's about 6 metres of small bowel inside there that's just chronically inflamed. And if you think of it that way, the child's just not going to be well, even if they look well. So the management of celiac disease. So it's, and we'll come to this again, it's a gluten-free diet and it has been for a very long time. So in the average Australian diet, there's around 20 grams of gluten per day, which is about 20,000 milligrams. In one slice of bread there's 3,000 milligrams, and we know that 50 milligrams a day of gluten is toxic to people with celiac disease. So that's like 1/100th of a piece of bread. So that's why when people go on a gluten-free diet, we are really clear with them that you cannot share butter, you cannot share the toaster, you must avidly avoid gluten altogether. But that treatment, as Yvette has said, is quite onerous. It's socially isolating. People do not like to turn up to restaurants and hassle the waiter about whether this is gluten-free or not. And it's usually unsuccessful. And studies have shown that 2 and 5 years after the commencement of a gluten-free diet, around 50% of people still have villus abnormalities. So it doesn't seem to work that well. We recently did a study here where we had celiac disease patients who were coming on to a trial. And these were strict people who thought they had really well-controlled celiac disease to go on the vaccine trial. They had negative serology when we saw them. They filled out a CDAT score, so a score less than 13 equates to very good control and avoidance of gluten. But we found 80% of them were not in remission. So we now talk about villus height crypt depth ratio, so the height of the villi compared to the length of the crypts. And 80% had a villus height crypt depth ratio of less than 2, and we think 2.5 is normal at a minimum. And non-healing of the bowel equates to an increased risk of cancer and osteoporosis. So there is a real unmet need for other therapies. And as you know, if you have celiac disease and you're on a gluten-free diet, there is no government rebate for that treatment. Yeah, so it's expensive. So this is a table from around 10 years ago, looking at new therapies for celiac disease. And you can see here that the peptide vaccination, or NextVax2, was in preclinical phases then for its trial development. And then this table's from 2015, and you can see now there's a lot more agents being trialled for celiac disease, but equally the vaccine had moved on to phase 1. So that was in 2015. And as of this year, it's now in phase 2. It commenced phase 2 trials around 2 months ago. So before a drug can get to the market, it needs to go through 3 phases of trials, phase 1, 2 and 3. When it's in phase 1, there's a 30% chance of success, but when it moves to phase 2, there's a 70% chance. And I know that with this vaccine trial, they have gone slow in their phase 1 trial development to go fast. They have worked out very clearly what the dose of this investigational product should be and how to use it, which we'll discuss. So what is NextVax2? What is this vaccination? And when we think about vaccinations, we traditionally think of them as something we give to prevent disease. So we give a vaccine to prevent measles. So it's changing the immune response. We use this vaccine for an established immune response. So the immune response in celiac disease is the upregulation of those T cells. And what we're trying to do is turn off those T cells through a vaccination. So it's sort of a reverse way of thinking of it. And originally I showed you that picture of that long gluten protein. So that's just one protein that's found in wheat. There are hundreds of gluten proteins. So what Bob Anderson and Jason Tai did, and I have to acknowledge Bob for passing on some of these slides to me, is they went back and categorised all the toxic hepatites in celiac disease, and they picked out the 3 that are most toxic. And they put them in one compound, and that's the injection we use for the celiac disease vaccination. Currently, it's only being tested in people that have a DQ2.5 genotype. So there's 2.2 and DQ8, but it's in 2.5, which accounts for over 90% of people with celiac disease. So before I go on just to discuss the trials that have been done, there's just a couple of things I wanted to say just to set the scene. So when people have celiac disease and they're diagnosed and they go on a strict gluten-free diet, generally their symptoms go away. Sometimes their symptoms then become worse if they're inadvertently exposed to gluten. So they may be on a gluten-free diet for 12 months, 2 years, and then they get a bit of a gluten exposure and they notice that their vomiting is profound and they didn't have that before. And when you do gluten challenges, the interesting thing is if you give those people gluten, you can almost time to the minute when they're going to start vomiting. It's usually about an hour and 40 minutes, and they'll tell you it's coming. The guy next to them will tell you it's coming. Everyone knows. And they vomit and they feel better and then off they go. So we've seen that for a long time, but we've never really understood why that happens. And it's probably due to this T cell activation, which has probably been shown in these trials while developing this vaccination. These phase 1 studies have also shown that this treatment is safe, it's tolerable, and it does suppress the T cell response. So again, back to our pathogenesis slide. So this is the gluten protein, and like I said, there's hundreds of those. The 3 most toxic epitopes have been defined. They've been put in a compound. And that's now injected. So the injected compound comes in and sits in this HLA molecule. And if you slowly increase the dose, you can turn off the release of interleukin-2 from these CD4 cells. If you ingest gluten and it's absorbed, it goes across the bowel wall, then a very important step occurs where it's deamidated. It takes on a very strong negative charge, and that changes the way it sits in this HLA molecule. So when you inject it, it's a slightly different route of administration. The other important thing is they're much the same. So we've always thought that with celiac disease, it's when you ingest gluten that you get sick. But what these studies have shown, you can replicate those symptoms by injecting the gluten epitopes as well. So this is what I was just talking about. This is a graph from one of the studies. I think it was the second one. And these people here are all on a strict gluten-free diet. And then they come in and they have a gluten challenge and they get symptomatic. That's generally when they vomit and get abdominal pain. And the reason why they had to have the gluten challenge for this study is we need to see that their CD4 cells were reactive. We wanted people whose immune system was reactive so we could shut it off. But when we gave them the NextVax 2, they developed similar symptoms. So NextVax 2 is very similar to the gluten epitope, so whether you take it orally or whether you get injected, these people are developing almost identical symptoms, which is fascinating, really, and attests to the systemic nature of celiac disease. And for the first time, it's been shown why we think that's occurring. So when you give people NextVax 2, what became obvious was after about 2 hours, you could start to measure interleukin-2. And that was the same response that was then shown in people when they were eating gluten. So interleukin-2 is one of those cytokines released from those T cells. Yes. And that holds a lot of promise for other reasons, because people who may not have celiac disease but are gluten intolerant may not be releasing interleukin-2. So it may be a new way of challenging people instead of the 6-week gluten challenge. And this is a spiderweb plot. So you can see this is the first dose of NextVax 2. These are all the cytokines that were measured. So you can see with the first dose, you get quite a vigorous response of interleukin-2. But by the 16th dose, there is barely any interleukin-2 being released. So the immune system has been turned off by this vaccination. This was the very first in-human trial, which was conducted in Melbourne and Brisbane. And the phase 1 trials were all about getting the dose right. So the dose was estimated to be 9 micrograms, or there was a cohort with 30 micrograms, or 60 micrograms, or 90 micrograms. So 6 people got 9 micrograms, 7 people got 90 micrograms. And we started to see that symptoms occurred when they got 60 or 90 micrograms. And one of the patients actually had to come off the trial because of those abdominal symptoms at that dose. But that's really important. So we started at a particular dose. We didn't up-titrate the dose. Yes. And this is the next 2 studies, the 1002 and 1003. And it was just really a difference in doses. So again, after the first study, we thought, well, 60, 90, 150 micrograms, that's probably the dose we're going to use for this vaccination. So that's what they got. These people got 3 doses over 3 weeks. These people got slightly different doses, but twice a week for 8 weeks. And what you can see again is this massive first dose effect when you gave the vaccination, and similarly some spikes thereafter. And again, this is the gluten challenge. And this is looking at the plasma cytokines. So when you give the first dose of NextVax 2, you can see in the red here cytokine release. But by the last dose, it's no longer there. So after those first 3 trials, Bob Anderson came up with the idea, well, maybe we're doing this a little incorrectly. So instead of giving people fixed doses, why don't we start really low and increase the dose to see if we can turn off the T cells as we're going. So with the 1004 trials, we did that. We started at 3 micrograms, and by doing that, we were able to show we're able to get up to 900 micrograms of the vaccine, whereas previously 150 micrograms had been problematic. And 900 micrograms in a vaccination is equivalent to around 2 loaves of bread. So the T cells are not reacting with a huge dose of gluten. And equally, the plasma cytokines, when you start with a small dose and you slowly increase it, you don't get that cytokine release either. So you are teaching the T cells not to react, which is the first time that's been shown in an autoimmune disease. So finally, with the celiac clinic and what we're trying to do at WMR, I think there's many opportunities for WMR to be involved in celiac research. And one is, of course, with NextVax-2. So NextVax-2 has now entered phase 2 trials. There's been 5 trials performed in Australia over the last 10 years. Queensland's been involved in every single one of them, and this one's coming to an end soon. And there will be a phase 2 rollout program in Australia. Over the last 10 years, we've been quite successful in enrolling patients into these trials. So there's been around 130 people who have now received NextVax-2, and around a quarter of them have come from Queensland. So we're very grateful for everyone that's put up their hand to be involved. But equally with that, it now gives us a real focus of experience with this product globally here in Queensland. A couple of the things we've done over the last 12 months I just wanted to point out that's happening upstairs. So remember I spoke about the HLA DQ2 and 8. So people will come in clinically, and if they're on a gluten-free diet, you want to know if they're at risk of celiac disease or not. So you do their gene test. And your genes should not change. They should be the same. So if I test them today, it should be the same as what it is in 2 years' time. If you don't have the gene for celiac disease, then you don't need to go and have that gluten challenge. But we were seeing people that came in that had, for all other reasons, celiac disease, but their HLA-DQ2 and DQ8 were negative, and we found that quite difficult to explain. So we did a little audit of our population and found that 9 out of 211 that had gene testing, for whatever reason, had them done at 2 labs. And with one lab, we found that patients were genetically susceptible for celiac disease when they actually had celiac disease, whereas in the other lab, it was reported as they had no susceptibility for celiac disease. So their gene test was incorrect. And we found that in a couple of people. But equally, it worked the other way, and we had a third reference laboratory as well that confirmed the correct one. But equally, we found that people who had normal celiac serology and didn't think they needed to go on to a gluten challenge or a gluten-free diet because they had negative HLA typing, in fact turned out to have positive HLA typing. So that small audit led on to a larger study, and we've since looked at 10 laboratories from around Australia, and Celiac Australia has published guidelines on the manner in which we should be reporting HLA typing. And we found that 80% of the laboratories that were looked at were underperforming in respect to the guidelines for HLA typing in terms of how they report their genotyping, their interpretation of it, and the methodology they use. So even though we have a vaccine that hopefully will become available soon, there is a lot of other research that still needs to be done for celiac disease and a lot of advocacy for people with celiac disease. This is a point of care study that we've been running for the last 12 months as well, and I'd really like to acknowledge the Thorson family for this, because without their support this simply would not have occurred. But it comes back to a vet's comment about children, and I think it's really important that we start to diagnose all the children with celiac disease so they can have all the opportunities of non-ill children. But taking a child off to get a blood test is a big deal for that child. They don't like to go to a pathology collection centre, they don't like to have a needle stuck in their arm. And equally for the parents to get them to the doctor, they have to go, get the form, convince them that they need it, get the test done and come back and see them. So it's fairly labour intensive. With celiac disease, we know, as the vet said, there is a long delay between presentation and diagnosis. In the US, the average time is between 9 and 11 years. And it's usually due to a delay in diagnosis, not to a delay in presentation. And as I said, in Australia, case ascertainment is exceptionally low. For a first world country, diagnosing 1 out of 5 kids with celiac disease, I just don't think is where we should be. So one way we can look for celiac disease is to go out and actively look for it. So relatives of people with celiac disease. There's not enough evidence to screen everyone for celiac disease, but certainly case ascertainment in high risk people is recommended. And the most predictive risk factor for celiac disease is a family history of celiac disease. If you are a first degree relative of someone with celiac disease, your risk of having that is around 7 to 8%. If you're a second degree relative, it's between 2 and 3%. The risk is higher in siblings of people with celiac disease, followed by their children, and followed by their parents, depending on who the person with celiac disease is. And interestingly, sisters and daughters, as we've seen in the prevalence of celiac disease, have a higher prevalence than their brothers and sons. So, the blood test for celiac disease, it's dependent on being on gluten at the time. And traditionally we take it with a venipuncture. But now there are point of care tests for celiac disease, so much like a pregnancy test. So people can come along, have a pinprick of blood removed from their finger, and we can determine if they have celiac disease or not. And there are quite a few of these point of care tests available. These are the two with probably the most research behind them, and they're the two we've been looking at here. To power this study, we need around 260 people, and so far I think we've recruited between 180 and 190 people, so well on our way. So with this study, we find a person with biopsy-confirmed celiac disease. We try and get their first-degree relatives to come in. We check their HLA typing, and then we perform a point-of-care test and send them off for a blood test. And we're seeing if the point-of-care test with the HLA typing is as good as a traditional blood test. And finally, the last thing that we're embarking upon here currently is to do with the gluten-free diet. And it may surprise people here tonight to realise that the gluten-free diet is not the same around the world. So if you live in Australia or New Zealand, we talk about parts per million. So if you're on a gluten-free diet, it doesn't mean you're actually on a completely gluten-free diet. Australian laws mandate that if you're selling a product that's labelled gluten-free, there should be no detectable gluten. And currently that's about 3 parts per million. So parts per million means that if you have a kilogram of gluten-free food, 1 milligram of it, if it's 1 part per million, will contain gluten. 1 milligram of it will be gluten. If it's 3 parts per million, 3 milligrams of that kilogram will be gluten. Whereas in the US and Europe, the guidelines are different. They allow 20 parts per million of gluten in their gluten-free food. And all of this really is based on one paper that looked at around 30 people. And what they found in that paper was that with 50 milligrams, people deteriorated, but they couldn't really say what happened with 20 parts per million. So 50 milligrams was seen as too high, but 10 milligrams, which equates to 20 parts per million, was allowed. Australia didn't adopt that, but recently is thought to do that because they think having such restrictive guidelines may limit the options of gluten-free food to people. So what we're doing is comparing 20 parts per million with no detectable gluten. And that's about to commence. We're doing that in Brisbane, Melbourne and New Zealand. So in summary, I hope I've explained some things tonight or triggered your interest. Really, what we did is we started talking about when celiac disease was really defined by Wilhelm Dickey. And before that, there was a mortality rate with celiac disease. We've talked about the prevalence and other features of celiac disease and how we diagnose it. And some exciting things happening in the world of therapies for celiac disease, a lot of which has occurred here in Australia. And finally, some things that we'd like to do here at Wesley. So I'd just like to thank you all for coming. And I think with celiac disease, the important thing is to thank the people that undertake the research. And so celiac disease research is not really being done in mouse models, it's being done in humans. So the results you're seeing tonight are from humans, whereas with a lot of other research, what we're seeing is from mouse models. So thank you very much.
Thank you, James. Every time I listen to James Davison, I learn something new, and I have the very great privilege of working alongside him. So every week we have a conversation. So every week I learn something new. I hope you found tonight to be informative. Certainly from the vet's presentation, we realised it's not just about celiac disease, it's about people living with celiac disease. And I know that everyone here has made the effort to come tonight for a real reason. And some of you might just be interested in celiac disease. I know that some of you have celiac disease. And I know that some of you have loved ones, friends, people you cherish with celiac disease. So I just want to share with you, we are the same. Within our very small family of Wesley Medical Research, we have a colleague with celiac disease, we have a partner with celiac disease, and we have a child with celiac disease. So this is not a theoretical application for us. This is deeply personal. As James has alluded, celiac disease is our focus, but it's part of a bigger spectrum of autoimmune disorders. And for the non-doctors here, the immune system is a very complex, dynamic system. But it holds the secret to so many things. When it works at its best, it protects us from invaders, whether it's infection or cancer. Sometimes it's underactive and makes us vulnerable to invaders. Sometimes it's overactive and gives us symptoms we don't want. And at the extreme end, it can actually attack our own bodies, leading to a whole range of different autoimmune disease. I think there's about 80 recognised so far. And the reason this research that we are doing is important is because while we are studying celiac disease, we are actually getting a better understanding of the immune system. And that understanding of the immune system is going to give us the keys to unlock the cures for everything from the common cold through to the most aggressive cancers. This is really important work. At WMR, we pride ourselves on being a very small organisation, but with very big outcomes. And as Professor Allen alluded, our research is specifically targeted to find real outcomes for real people in real time, and by real time we're looking for answers within 5 years. This is why it's so exciting for us to work alongside James. Celiac disease is common, we can help many people, and the work that we're doing, because of his international reputation, the work that we are doing as a small organisation is going to have a big impact for the people in our lives, the people we cherish and the people that you cherish. I think it's fair to say that probably every person in this room right now wants better, easier, more reliable, more accurate diagnoses. We want better treatments, and ultimately we're looking for a cure. And even when we find that cure, we will then generalise that knowledge to take on other autoimmune diseases. You may not have noticed, but as you walked in this afternoon, you would have walked past the famous words of John Wesley, and let me share them with you because I think it's a timely reminder. John Wesley said, "Do all the good you can do, by all the means you can, in all the places you can, at all the times you can, to all the people you can, as long as you can." We live by those values, and we invite you to join us in our community of people who are trying to do the most good for the most people in every way that we can. How can you potentially help us further the cause of James' work and the work of Wesley Medical Research? There are many ways, in the means that you can, as John Wesley said. The first and simplest is just merely by raising awareness. Please talk to your friends, your families, your colleagues. Please spread the word. Please join our Facebook site and watch for updates. Please join us on LinkedIn. Some of you may even go the extra step and potentially put your bodies on the line with us. So obviously we are looking for people to help us with our clinical trials. And so if you are interested or you know people who might be interested, please register. We're keeping a collection of names, so as trials potentially unfold, we can contact people. And finally, obviously, we need donations to help us pursue this very important work. And the better funded we are, obviously, the better we can do, the more we can do, and the more people that we can help. Thank you for being with us this evening. I'd like to invite you to join me in thanking our special guests and speakers, Professor John Allen, Yvette, and James Davison. On your seats is a take-home card, and don't forget to log in and have a look at our website. And finally, I'd like you to join us for refreshments, and I want you to know that all of the refreshments tonight, organised by the lovely Sarah Hutton, are all completely gluten-free, so they are very safe for our entire audience. Thank you.